Discovering how the genetic code works was no easy endeavor, but there were a few people brave enough to take on the challenge. Two of these people were Marshall Nierenberg and Heinrich Matthaei. At this point in history, it was already determined that the genetic code was a non-overlapping code that contained codons consisting of 3 nucleotides. So in each strand of DNA, 3 adjacent nucleotides coded for a certain amino acid, and each of these nucleotides were part of only one codon. With that information, Nierenberg and Matthaei set out to figure out what amino acids were specified by only one type of base. So basically, they wanted to know, what does the codon UUU or AAA code for? So to do this, they took all the components of a cell that is necessary for the translation process and put it in a tube. The only thing this mixture was missing was mRNA. They then had artificial mRNA made that only contained one nucleotide. The first one they did contained only uracils. This is where the experiment gets a little tricky. They then had 20 different mixtures of amino acids. Each flask had a different amino acid that was labeled radioactive. The cell components mixture and the U-only mRNA was added to each mixture, and the form protein was then isolated. Only one of the 20 isolated proteins came up radioactive. This was the flask that had phenylalanine as the radioactive amino acid. Nierenberg and Matthaei then concluded that the codon UUU coded for phenylalanine. They repeated the process with A-only mRNA, C-only mRNA, and G-only mRNA. They discovered AAA codes for lysine, CCC codes for proline, and they were never quite able to get GGG to work. After this, Nierenberg decided to take the next step with Philip Letter to discover more of the genetic code. They wanted to know what other codon and amino acid pairs could be determined. To do this, they used a 3-nucleotide-long mRNA strand with known codons plus ribosomes, tRNA, and the 20 amino acids, one of which was marked radioactive. The mixture was then poured over special filter paper that binds to ribosomes. The ribosomes bind to the mRNA, tRNA, and its amino acid, and this is what remains on the filter paper. Everything else then filters out. Nierenberg and Letter then checked to see if the paper was radioactive, and if it was, then that codon sequence coded for the pre-marked radioactive amino acid. Similar to Nierenberg and Matthaei's experience, they had to do this 20 times with different amino acids marked as radioactive. For example, when mRNA with the code GUU was used, they discovered a radioactive filter with valine as the marked amino acid. Through this, Nierenberg and Letter were able to identify 61 codons, and therefore making another huge step in learning how each of us were uniquely made.